Hi everyone, Dr. Linda here. In this short video, we're going to be going over the organelles and the different parts of the human cell. And this PDF just has about, looks like five slides to it. Okay, so let's zoom in and let's take a look at some of these structures. So here, right on the top, we see the cell membrane. We went over that it is a phospholipid bilayer. We know that the outside of the cell here is hydrophilic, and the inner part of the cell membrane wall is also hydrophilic, and right in between is hydrophobic. That's where the saturated and the unsaturated lipids or fats are located. Outside of the cell is extracellular fluid, and inside of the cell is intracellular fluid, also known as the cytosol. Okay, but when we're talking about everything from inside of the cell membrane to the nucleus, that's called the cytoplasm. Okay, so here on the left, we see the cytosol. That's just the intracellular fluid, but the cytoplasm is everything from the inside of that cell membrane to the nucleus. Okay, let's see what else we have here. Let's look at the very center of the cell. We have the nucleolus. The translucent clear part that's superficial to it is the nucleus. And then superficial to the nucleus is the nuclear envelope. And then it says the nuclear pores are the holes in the nuclear envelope. So these are nuclear pores. Again, nuclear envelope is the white, nuclear pores are the holes. The clear section here is the nucleus, and the nucleolus is the orange structure contained deep within. Continuous with the nuclear envelope is the endoplasmic reticulum, but in this case, it has the fixed ribosomes. So it's called rough ER, or the rough endoplasmic reticulum. We could see here, rough endoplasmic reticulum are the white dots, which are the ribosomes. So this is rough endoplasmic reticulum, but right here, this would be smooth, right? There's no ribosomes here, or even here. This looks smooth, looks very different than this, right? So down here, this is rough endoplasmic reticulum, but here there are no fixed ribosomes, so it's smooth endoplasmic reticulum. Here, these the stack of pancakes that we see here, the orange package right there, boom, boom, boom. That's the Golgi apparatus. Remember, the Golgi apparatus repackages proteins, and it pushes out the secretory vessels. So this is a secretory vessel, a secretory vessel, all of these were released or secreted out by the Golgi apparatus. Okay, and maybe one of the things that it can do is that it can push out one of the secretory vessels, could be this right here. See, it's called a secretory vessel. Looks like that light bulb, and it kind of opens up to the cell membrane, and it says exocytosis or endocytosis. So exocytosis is this vessel's ability to release its contents outside of the cell or to become part of the cell membrane wall. Or endocytosis is the ability of bringing and drawing something in to the cell, right? Endo is in, exo is like exit out. So endocytosis, bringing something into the cell, exocytosis, removing something from the cell. But this vesicle started out as a secretory vessel. But the fact that it moved towards the cell membrane wall, it makes it either exocytosis or endocytosis. And unless I tell you, hey, this is releasing something from the cell, that would tell you exocytosis. If I said it's bringing something within or into the cell, then it's endocytosis. Okay. Here is the black structure going around is the centrosome with the yellow microtubules. That's called the centrioles. Centrioles, centrioles. The black sphere is the centrosome. Down at the bottom at 6 o'clock, that's the lysosome that contains the digestive enzymes. The orange here, these are mitochondria. This is the mitochondria cut, and this is the mitochondria whole. This is allowing you to see what the matrix within the mitochondria. The gray is the peroxisome that's right here. Okay, I think we covered them on this one. Let's make sure. Looks like we did. Let's see if we can zoom in here and look at the other structures. So this is zooming in at around the 11 o'clock position, right? So we're focusing in 10, 11 o'clock, what we have here. So this is the mitochondria. This is the Golgi apparatus with a secretory vessel. This is a peroxisome. And this is also a secretory vessel that is releasing its contents. That would be exocytosis. Here's the cytosol, the clear fluid within the cell. Here is the cell membrane. The black sphere is the centrosome, along with the centrioles. We have ribosomes that are fixated to the endoplasmic reticulum, which makes it the rough endoplasmic reticulum. Here is the clear liquid called the cytosol. The orange pit is the nucleolus. The clear translucent part is the nucleus. The white part is the nuclear envelope with the nuclear pores within it. The yellow at 6 o'clock position is the lysosomes. The lysosomes clear cellular debris. It's got hydrogen in it and digestive enzymes to assist. Here's another view of the rough endoplasmic reticulum with the white ribosomes fixated to it. And we contrast that to the smooth endoplasmic reticulum without the ribosomes attached to it.